Hello and welcome to Agri History. In this episode, we'll cover the topic of lentils. Lentils are part of the genus Lens, which is a small but genetically diverse genus. This genus is part of the same family as all other legumes, the legume family. The scientific name for the cultivated lentil is called Lens culinaris, but there are many other different species of lens, such as Lens ovoides, Lens montbreti, Lens nigricans, and Lens orientalis. The cultivated version of Lens was domesticated in the Fertile Crescent, along with many other founder crops, and its wild progenitor has a range from Greece to Uzbekistan and the Crimean Peninsula all the way to Jordan. In this video, we'll cover the origins and domestication of lentil. The oldest remains of fossilized lentils are found in a Greek cave, and the samples were dated to 11,000 BC. There is also a site in Syria with samples of lentil dated between 8500 BC and 7500 BC. Unfortunately, we cannot distinguish the state of domestication, if it was domesticated at that time, from these samples. But the remains of lentils found in an old village site had a large storage of lentils dated back to 6800 BC. This indicates the minimum age of which lentils were cultivated. The earliest signs of fully domesticated lentils were found in a site in Iran, which were dated between 5500 BC and 5000 BC. These lentils had larger seeds than the wild lentil, and as such, these samples were probably domesticated. This is how we confirmed that lentils were domesticated within this area of the Near East. This was further confirmed via the fact that most samples of agriculture were the remains of lentils. Based on the data found in these sites, it is likely that both wild and domesticated lentil were consumed in that area. Lentils were introduced to southeastern Europe, and through the Danube River, lentils came to Central Europe. Based on its archaeological evidence, lentils were likely domesticated by the 5th millennium BC. Lentil seeds were likely spread across the Nile Valley in Neolithic times, and during different wars were spread to Ethiopia. On the eastern front, lentils spread to Georgia in the 5th and early 4th millennia BC, and appeared in India and Pakistan between 2500 BC and 2000 BC. Lentils are also found among the antiques of ancient Egypt. All Egyptian documents mention lentils, and these documents were written in the 12th dynasty, which took place between 1167 BC and 2000 BC. Theophrastus wrote about lentil cultivation methods, and from numerous classical sources we learned lentils were a primary food source for the poor of ancient Greece. One of the more famous agricultural philosophers of ancient Rome, Moderatus Columella, wrote in his book De Re Rustica, wrote down the cultivation practices for lentil, which included harvesting, seed conservation, and the prevention of weevil damage. Lentils were also used as ritual offerings for Roman graves. The name lens came from the Greek word lens, which means lens, because the seed looks like a lens, if you look at it and squint, of course. Based on genetic and morphological evidence, it is likely that the cultivated lentil evolved from Lens orientalis. Experiments with crossbreeding Lens orientalis with our cultivated lentils have shown that the differences between our cultivated lentils and the wild species can be traced to at least 6 genes. The SCP gene in the wild species codes for brown seed coatings with dark spots. A mutated version of that gene is found in lentils. This causes a yellowish-gray coloration on the seed coat. This mutation is likely recessive because when the two species are crossed together, the progeny have the seed coat of the wild species. The GH gene in the wild species produces prostrate growth habits for the plant. The mutated gene in regular lentil produces bushy growth, and the progeny of the crossbreed of both the wild and domesticated species bred progeny with a mix of both traits, indicating a single gene with incomplete dominance. Flower color is controlled by a single gene, as is the epicaudal color. The gene that prevents shattering is a recessive version of the shattering gene in the wild lentil. The hard seed coat in wild lentil is also controlled by a single recessive gene. This showcases that Lens culinaris, or common lentil, evolved from Lens orientalis. In seed crops, there is a process called domestication syndrome. This refers to the different changes that occur when a plant goes from being wild to domesticated. These changes include, but are not limited to, seed size, seed pod shattering loss, seed dormancy loss, giganticism, and changes in the plant architecture. As we previously discussed, seed pod shattering and seed dormancy are controlled by single recessive genes, but the major trait of seed size seems to be controlled by more complex genetics. There are several hypotheses for how lentils were domesticated. The first one's called the weed/dung heap hypothesis. In this view, people accidentally brought these seeds over to their locations and dumped them here and there, where they became a little bit weedy. Thanks to better fertility within the human habitations, the best plants would be found and selected. This would trigger the idea of cultivation, and these early farmers would save the seed of the plants that had the easiest cultivation capabilities. The second hypothesis is that lentils started off as a weed or secondary crop that got mixed into a pre-existing cultivated crop like wheat, and under certain conditions, lentils would thrive and be consumed. This would lead to cultivation and eventually domestication. A third view is that individual tribes would offer seeds of lentil for ritualistic purposes. On these areas, these seeds would wind up growing in high densities, allowing them to compete with weeds and be susceptible to cultivation later on. None of these have been confirmed as of yet.